So this is a diploid cell that has already gone through S phase and is about to go through meiosis. So they already have, the chromosomes have already duplicated, and so we have the sister chromatids that are duplicates, basically exact duplicates of each other, next to each other, and the centromeres, our imaginary centromeres, are what are holding them together. So yeah, this is the beginning of meiosis 1. Woo. So in prophase 1, the nuclear envelope fragments and the homologous chromosomes, they line up like this. They go through the synapses, which is basically when the two pairs of homologous chromosomes like come together like this, and they also at this point go through the crossing over. Well, this is a tetrad. The synapse forms a tetrad. They go through the crossing over, which is basically when some of the DNA from each of them, because they're so close together, switches, like so. And this happens for genetic variants because that's always great. And another thing that also helps with the genetic variants here is that they're an independent orientation. So the homologous chromosome pairs don't necessarily have to be like this. They could be like that, or they could be like the other way around. So you get, and especially as like the more chromosome pairs there are, the more variants you get. So that's great.
So during metaphase 1, the pairs of homologous chromosomes start to move towards the center, and the spindle fibers attach. Sorry, my head's in the way. The spindle fibers attach to the homologous chromosomes. And then they during anaphase, the spindle fibers start to pull the homologous chromosomes apart, and the spindle fibers coil up. And then during telophase, the nuclear envelope starts to reform. The fragments start to reform and elongate. And then nuclear envelopes reforms and elongates. And then during cytokinesis, the cleavage furrow, the cleavage furrow for the cell wall.
Can we pause it?
So this is a centrosome in the whole thing, and each individual one is a centriole. And during meiosis 2, the same thing happens in meiosis 1, except it occurs with the sister chromatids. And in prophase 2, the nucleus starts to fade away and the spindle fibers start to elongate in the cell. And in metaphase 2, the sister chromatids start lining up and connecting to the spindle fibers. In anaphase 2, the sister chromatids start to separate and go towards the centrosome, and the spindle fibers start to shorten, and the cell starts to elongate. And in telophase 2 and cytokinesis, the cells become 2, and the nucleus starts to reform in the new cells. And instead of being diploid cells, they are now 4 separate haploid cells.
So also, just a note, these are considered haploids as opposed to diploids because they only have one set of chromosomes. They don't have homologous chromosomes.